turns out that, that it's the violations of these. It turns out when you find that this equation doesn't hold, that you can actually get information about for new mutations or selection at certain sites, or about the migration of people from one place in the world to another. So it's actually when this doesn't hold, there are lots of interesting things that can be discovered. Okay. So just to show you how this works and how that there are actually differences in the allele frequencies and genotype frequencies in different populations. I won't go over everything in this list, but let's just look at one. This is sickle cell anemia that we talked about last week. And I mentioned that this, this liability allele, for some reason, the people who are heterozygous for this have one normal hemoglobin beta chain and the other one that's mutant. People who are heterozygous seem to have resistance to malaria. So this mutant allele the frequencies are much higher in regions of the world where malaria is a problem, and particularly in equatorial Africa, this seems to be the case. And so if you look at African Americans, many of whom came from originally, their ancestors came from equatorial Africa, you find the frequency of the allele and the frequency of homozygotes is much greater than, say, for example, Hispanic Americans or Europeans here. Oh no, I guess that's a different disease. But if you looked at the Caucasian population, the frequency would be much lower than in the African American population. For other diseases, you find that's just the opposite. So, for example, cystic fibrosis, as we talked about, may provide resistance to certain diseases. And the cystic fibrosis mutants are found at very high frequencies in the Ashkenazim, or the Ashkenazi Jewish community from Central Europe, where they're not found in other populations. So depending upon the population, the frequency of a certain allele can be vary dramatically, and and along with that, the prevalence of the disease can vary. Now we talked. Okay. So the next thing I want to talk about is how you map genes to certain chromosomes, or to specific places on the chromosome. And this is a pretty complicated business, and so I don't have time today to go into all the details of this. But what I will do is introduce you to certain important concepts, and it'll sort of give you a basis for understanding more complicated topics. Now, the key to understanding where genes are located is this concept of linkage. So we're going to talk about that first. So in order to find out where a disease locus, a disease gene is located on a chromosome, you need to have some sort of marker. Now, if we talked in the first lecture about recombination between chromosomes and the fact that at every generation, during meiosis 1, you have the pairs of chromosomes lined up, line up at the plate of cell division, the plane of cell division. And actually before they line up, they form, you have synapses where homologous chromosomes make this complex with each other. And during that time, this is during prophase actually, a recombination takes place. Then they line up at the plane of division, and they start to pull apart from each other, and you see that those chiasmata between the two pairs of chromosomes. And those are places where recombination is taking place. Now, in every generation, every chromosome undergoes at least one, often 2 or 3 or 4, recombinant events. And those are very important for, if that didn't take place, we wouldn't be able to map genes very carefully. So let's think about the case at first where there's no recombination, and what that would mean for being able to map genes. Well, if there's no recombination, that means that essentially every chromosome may have a marker on it, And the best you could ever do would be to map a gene to a particular chromosome. The best you could ever do is to show an association between a marker in chromosome 1 and this disease. But you would never be able to tell where the gene is located inside chromosome 1. So so let's consider then, let's look at this diagram and sort of consider the case where you don't have recombination taking place. Well then, on chromosome 1, for example, you might have a marker for that chromosome. This is, I'm talking about chromosome 2. This is, say, chromosome 1, and this is another copy of chromosome 1, a different copy of it. And suppose this marker consisted of suppose it was a SNP, and suppose there was a C allele here and a T allele here. So this big M would be C and this little m would be T. Okay, so you could distinguish those markers. And suppose that the disease allele was associated with this C marker. Well then, if there's no recombination, then this C allele here, the big M, would always be associated with a disease SNP. So that when the gametes are formed from these, you could get only this one can give rise to this pair of gametes, and this one can give rise to this pair of gametes, and that's all the possible gametes that you have. Okay. So then if you looked in the progeny, and if this were a dominant mutation, say, you would always find that the disease would be associated with this marker, the C allele, and it would not be associated with this little marker, the T allele. Okay, does that make sense? So, but because there's recombination, it's possible that this marker would not always be associated with disease. So this C allele here, if there's been recombination between the two, would not, in this case, be associated with disease. It would be instead this would be associated with the disease. Okay, so the fact that if there was no recombination, the best you could ever do would be to map a disease to a particular chromosome, and that's it. If you had one marker, or any marker for chromosome 1 would be equally good, because there's no recombination, and that a marker would always be associated with that chromosome. And if you had 23, or 24, I guess, markers for the 22 autosomal chromosomes, and then the X and Y chromosomes, then you could, in theory, find the association of a disease with a certain chromosome. But because recombination takes place, it is in principle possible to find where in the chromosome a certain disease gene is located. Now the key to this is that, roughly speaking, the probability of a marker being separated from a disease-causing allele depends on the distance of the marker and the disease-causing allele. So for example, if a marker is, you pick a marker at one end of the chromosome and on say the P arm of the chromosome, and the disease-causing allele is at the other end of the Q arm of the chromosome, there's almost certain to be recombination between those two. And so in general, you would not find a correlation between this marker up here and the disease locus down here. Instead, you'd need to pick a marker that's fairly closely localized to that place where the disease-causing allele is. And then, and only then, you'd be able to find this tight association. So roughly speaking, there's a correlation between the distance between the marker and the disease-causing locus. And so if you have many markers along the chromosome and then check each marker to look for its association with the disease, if you find the marker, when you find a marker that is closely associated with the disease, so it's not disrupted by recombination, then you can say, well, the disease locus must be close to that marker. Okay, does that make sense? So that's basically the principle for genetic mapping. You start with a lot of markers, and you look at a lot of different individuals who've had recombination take place at different places. And then you look at all those individuals and you say, well, which marker is most closely associated with this disease? And then if you know where the position of the markers are within the chromosome, you can map the disease to that locus. So that's basically the principle. Okay, now in practice, this is done in two different ways. One is to look at linkage within families, within pedigrees. And this is actually the first type of mapping, genetic mapping, that was done and led to the discovery of many important disease genes. For example, Huntington's disease was found this way. So you start out with very extensive families that have where a disease is going through the family, and if you have a bunch of those pedigrees, by looking at and if you have molecular markers for all the chromosomes, looking at all the patterns of association of the markers with the disease will, in theory, allow you to say where on which chromosome that disease gene is located. Okay, so this approach, this linkage analysis, is done within multiple extended families. Traditionally, the type of markers that are used are those SNPs or mini or microsatellites. Unfortunately, though, even if you have fairly extended families that you're tracing the disease through, and even if you have many markers along the chromosome, the number of recombination events within any given pedigree is small. Again, every chromosome only undergoes several recombination events, for each mitosis. So even if you have several generations, there are not many opportunities to separate a marker from the disease gene. And even if you have many pedigrees, it's still not that many recombinant events. So for this reason, the best you can usually do by this approach is to say, well, this disease-causing allele is located somewhere within 1 million base pairs or 2 million base pairs of this marker. And that's about as close as you can get by this approach. And so, for example, when the Huntington's disease gene was first cloned, or first mapped in this way, it took, this was done by looking at an extensive pedigree in Venezuela, where the disease, Huntington's disease, had been passed from generation to generation, and they looked at many different individuals in this very large pedigree. Even then, they only knew within a few million base pairs where the disease gene was located. It took another 10 years of research to figure out what gene was actually causing Huntington's disease. So it gets you to a, it's better than not knowing anything, but still it leaves you with a large stretch of DNA to search for the actual disease causing mutation. So often additional mapping is required to identify the disease gene. And again, for Huntington's disease, this took 10 years to do. And then this is sort of a technical point, but some genes actually are stretched over very large regions of the chromosome. So some of the largest genes in the genome are actually stretched over 1 million base pairs long. And what that means is that you have exon regions that encode DNA, the sequences of proteins that are separated by very long introns, so that the whole gene stretches over a very long distance. And what happens is, for those genes, you actually have to transcribe 1 million base pairs of RNA to make the initial transcript, and then that RNA is eventually spliced to give you the continuous reading frame that you find in the messenger RNA. There are many genes like that, and so there's one advantage of this approach is that if you have mutations, say in this exon 1, or in the middle or in the last exon, any one of which can cause the disease, the fact that you're locating the disease within very large stretches of DNA turns out to be an advantage, because all these different mutations sort of map together. They're found together very often. So this method is actually perhaps more efficient than the method I'm going to mention next for finding disease genes that are very long. Okay. So here's an example. So this is looking at, this is supposed to be retinitis pigmentosum causing gene, which is, you remember from last week, is a dominantly inherited disease. And so the idea is if you have two markers, one here and one here, you find, and then you look at the correlation between the disease. So this is dominant. So if anyone who has this allele will have the disease, people who don't have that allele don't have the disease. If you look at then the correlation between the markers and the disease, you find that the correlation is excellent between this big B and the disease. So whenever you find big B, you find the disease. If that's true for this person. Here, little b is not correlated with the disease. Here, big B is correlated with the disease. So